next up, it is my pleasure to introduce Erich Huang from both Duke Institute for Genome Sciences and Policy, but primarily at Sage Bionetworks, who will be talking about clearScience, effacing the boundary between doing and communicating science.
Thank you, Marianne. I'm going to swap out the machine here with since my animation wasn't working on the other one. And I see I inserted a slide there, but we'll just skip past that. So I'm coming and talking from the standpoint of being actually a trained clinician, a trained wet bench scientist, and a genomic computationalist. And coming to Sage Bionetworks up in Seattle, I saw an opportunity to do something, and I'm going to talk about that, and it's called clearScience. And I'm going to start with, I don't know what's going on here. Sorry. I guess I inserted a bunch of slides. I'm going to start with a place that I worked very frequently, which was the examination room. I worked as a breast cancer surgeon at Duke, and I often met my patients in a room such as this, probably not quite as nice. And you have to do a lot in an examination room. When you make a treatment decision for a cancer patient, you're summarizing decades of information and making a decision based on decades of information that actually has life and death consequences for the patient. And for me, that's why doing something like thinking about reproducibility and transparency and thinking about the scientific method in general is very important, because I have to make decisions and trust that all these decisions that have been made before me have real scientific basis. So here's a patient that I saw in my exam room. That's not her real name, but Betty Jones. And she came to me and said, she'd previously been seen by the previous surgeon on my service, and this was a couple years later, and she came and said, I have a lump near my mastectomy scar. And that always gets you worried. The first thing you worry about in such a case is whether someone might actually have a chest wall recurrence. And again, you are dependent on everything that's been done before you to make the right decision for that patient. So here was this patient that I had. She was young, she was African American. She ultimately, by biopsy, was shown to have a triple negative recurrent cancer. And previously, when she'd had her previous surgery, it was a multifocal cancer. So these were all minuses on the tally for her. The only positive for her from her previous surgery was that she had lymph node negative disease, at least by that previous surgery. So with the possibility of a chest wall recurrence, and in fact that was biopsy proven, we elected to pursue an aggressive therapeutic strategy. This woman is only 36 years old. She had a lot of life in front of her, and she had an aggressive cancer. We needed to do the maximum we could do for her. And so how do we deal with this patient? And what we have as options for a patient such as this with a triple negative breast cancer are these. We have the standard stuff, standard chemotherapy, anthracyclines, taxanes, antimetabolites. We have standard platinum agents, so that's conventional chemotherapy. And nowadays we have targeted agents such as cetuximab, Effacing, other kinds of targeted inhibitors. So this is the new stuff. But the problem is, current knowledge is that all these therapies are a wash. So how do we proceed in this case? Because there are many reports that are very anecdotal about how we might potentially treat this patient, but it's hard for me to make a decision about how to treat this patient based on the current level of science. So I have this patient in my exam room, and I need to boil down 30 years worth of oncology and surgical knowledge to make a decision for this patient. And I have to stand on the shoulders of giants. And I have to trust that those people who preceded me are actually indeed giants. I love this quote from Isaac Newton, because this is probably the scientific mind of a millennium. And here he is in a letter to Hooke saying, I've seen further because I'm standing on the shoulders of my predecessors. And when I was faced with this patient, I'm faced with a similar challenge in that previously at Duke, one of my colleagues had published this paper called Genomic Signatures to Guide the Use of Chemotherapeutics, a high impact paper in Nature Medicine. This paper was cited 463 times as of my last check a couple weeks ago. And actually at Duke, it spawned 3 genomics-guided clinical trials. So we actually were using this on real patients in a real clinical trial setting. The problem is, someone quoted Begley earlier in the previous session, was that there are questions about how reliable this model was for personalizing cancer therapy. And indeed, it turned out at Duke, I'm sure many of you are familiar with it, was that this was actual scientific misconduct, concealed amongst millions of variables. It ended up in a 60-minute piece just a few months ago. So what we had here were millions of data points, a Nature Medicine paper which gave it a stamp of approval for a molecularly customized cancer treatment plan. But really, when it came down to it, what we're looking at is a black box. And to me, a black box is not science. It's magic. And we don't practice magic, we practice science. And certainly, from my context, if I'm making a clinical decision, I have to trust the science, and I do not want to trust a black box. So after that situation, the National Academies initiated, and the Institute of Medicine initiated a sort of forensic study and recommendations for how we go forth, and that led to this report. And what that report basically said, and it didn't really have lots of specifics, a lot of it's just common sense, but what they said was things have to be more open. But what does open really mean? So we have open source, we have open data, we have open access. So these are all good things. I definitely believe in these things. But does all that access really make things accessible? And when we have open source, open data, open access, is the science that underlies all these things actually really very clear to us? So here's a blog I like quoting. This is Bruce Booth at Atlas Ventures. He blogged this last spring, and he was blogging about how his venture capital firm makes decisions about their investments. And one thing he noted was that their rule of thumb at Atlas Ventures was that 50% of Cell, Nature, Science, PNAS papers are not reproducible. We heard about the Begley Ellis paper. We know that only 10% of landmark papers in cancer biology are reproducible. And in one of my postdoc studies of a certain TCGA paper, what he found was with almost 300 million data points, 124 pages of supplemental material, unknown lines of unobtainable source code, unknown version of or architecture of R, an R package which was previously web accessible but no longer is web accessible, this paper was cited 580 times, and we could not actually reproduce the results. So I think the problem is people think of scientific process as a one and done process. People define a question, they gather information, they form a hypothesis, they test their hypothesis, they analyze their experimental data, draw conclusions, and they publish. But the problem is it's not one and done. I think of it as a continuous process where it has to have a life that goes beyond that publication, that there has to be continual validation, forking, if you want to use a software engineering term, readaptation of science. So here's the typical scientific process, at least the way I see it right now. You generate your data. If we're doing more computational type science, you store it on a local server. You put it on, you analyze it on your local machine. You write a document. You submit that document to a journal. It's sent to the reviewer as a PDF by most of the manuscript submission systems. They accept it. They digitally typeset it, so it's printed on physical paper. You get a static HTML representation on the journal website, and you get yet another static PDF representation of your paper. You all know this. So here's the problem here. What I see is that there's a line separating the actual science from the scientific claim. And it's ridiculous. So what I think we should be doing is effacing that boundary. So with this clearScience project, which is funded by the Sloan Foundation, we want to reimagine scientific communication, make every component of a data-intensive project, let's say in this case a genomic analysis, web accessible, allow consumption of the content at a variety of levels of complexity and abstraction, and leverage open RESTful APIs. So we want to basically provide a way for the users to reconstitute the entire analytical environment in which they did their science. So that environment includes hardware, includes software, data, and code. And what we want to do, being based in Seattle, we have access. The great thing about Sage Bionetworks is we actually have dedicated software engineers, many of whom are people from places like Amazon. And we want to put this all in the cloud. So we want to put it in the cloud and create a narrative that coherently interweaves the scientific assertions with executable data, code, software, and hardware. The hardware is virtualized, obviously, that support them. And basically, at least in this first pass, what we're thinking about is thinking of my computer's a cloud, let me hand it to you. And we want to leverage APIs both from our proprietary Synapse. Oh, am I done already? Jeez. Okay, I'll stop. Well, I have a video demo of some stuff, but I'll stop. Yeah.
But know that you're there. Okay. Sorry about that.
That's okay. That's my fault for taking so long. Do you want me to plug that in?
Oh, is that it?